Oh god. Sorry, got to shut off the music. Okay, so today we're going to learn or for some of you on Tuesday, depending upon if you're staying after school. Today we're going to learn about gel electrophoresis. And the purpose of gel electrophoresis, or it allows us to sort or visualize DNA. If you remember when we learned about PCR, the purpose of PCR is to replicate lots and lots of DNA, or one segment of DNA, or a specific sequence of DNA by designing primers correctly. You need to design the primers correctly so they will anneal or stick to the space of DNA or the segment of DNA you want to replicate. And if it sticks, it will then replicate that specific fragment of DNA as long as you've designed the primers specifically enough so it only binds to one place in the DNA. But the question is, and sort of something that I overlooked while we were talking about it, let's say you run the PCR. Okay, the question is, how do you know you got any DNA? I mean, you can't see DNA with the naked eye. So how do you know whether or not your PCR worked? And the answer is, and what scientists will say, is that you run it on a gel. And the process sort of for sorting DNA and visualizing DNA is called gel electropheresis. So this lesson is going to go through how we can use gel electropheresis to both visualize and sort DNA, and then we're going to go through the purposes of this, what are some uses of gel electropheresis in real life. So we discussed before that you can use restriction enzymes to cut DNA, and restriction enzymes cut at specific sites, and we use them, or we discussed them when we were talking about bacteria transformation, when you cut the plasmid with one restriction enzyme and the gene that you want to insert into the plasmid with the same restriction enzyme, and that creates sticky ends, which I feel like we don't get truly. But we'll see when we start reviewing for the TIS. And because you're cutting them with the same restriction enzyme, they leave the same sticky ends, and then the plasmid and the gene will be able to anneal because they have complementary sticky ends. Restriction enzymes can also be used, and we're going to talk about how in this lesson, to just cut up different people's DNA. So everybody has sort of a DNA sequence that has sites. And if you cut it with restriction enzymes, because people's DNA is different, and we'll talk about specifically about where it's the most different, you can compare it. So you take DNA from one person and DNA from another person and cut it with restriction enzymes. And because their sequences are different, because we have different DNA sequences, and we'll talk about where in the genome this happens in particular, it will cut differently. And then you can use gel electropheresis to visualize the different cuts or the different strands of DNA you got from each person's DNA. This is used in forensics. It's used to diagnose disease. It's used to determine paternity, and we're not going to talk about this in this lesson. We'll talk about this when we get to evolution. It's also used to investigate evolutionary relationships between organisms. But first we're going to talk about how gel electropheresis works. So how can you visualize DNA fragments that have been cut with restriction enzymes? And the answer is gel electropheresis. So gel electropheresis separates DNA fragments by size. Okay, and the gel itself is made up of a gelatin. I think it's from. I'm actually not sure of the organism right now, but it's from an organism. It's a protein. It's from algae. Haha, there it is. And it has tiny holes in it, so it's a gel. And you'll see these gels on Wednesday. On Wednesday, you're going to practice what is known as loading a gel. And inside the gel, you can't see these. There are tiny holes that allow the DNA to travel down the gel. Because there are holes, okay, these smaller fragments are able to travel further down the gel, and the larger fragments can't travel as far. So if you look at this gel that we have right here, and you can see the sort of darker fragments, and I lost my pen here. These fragments here, so this is a strand of DNA, or when there's a dark spot, that means there's DNA there. The strands that are higher up on the gel are longer, or have more base pairs than the bands that are further down on the gel. They have less base pairs, and they're able to travel further because they're smaller and they can fit through the holes more easily. Okay. So now the question is, what causes the DNA to move down the gel? So this is sort of a picture of a gel right here. And this is, so here's the gel. And this is where you would load the DNA in this well right here. The DNA is negatively charged. And the reason why DNA is negatively charged is it has a phosphate group. Remember this phosphate group, so phosphate, this is the sugar, it's DNA, so it's deoxyribose, and this is the nitrogen base. The phosphate group is negatively charged because of the phosphate group here, so that makes DNA overall negatively charged. And you have this gel, which you'll see on Wednesday, and you hook it up to an electrical box that sends an electrical charge down the gel. The top of the gel gets the negative charge, and the bottom of the gel gets the positive charge. And because there's an electrical current running through the gel, it causes the DNA, which is pipetted into this well, and we're going to practice that on Wednesday, so there's DNA in here. It causes the DNA to migrate down the gel. So you'll sort of see the DNA migrating through the gel. Because there are holes in the gel, okay, the longer sequences, like this one, they don't go as far. So longer base pairs, they don't travel as far, they get caught in the holes in the gel. And the shorter base pair sequences, they get to travel faster, and they go all the way down the gel. So just to summarize, gel electropheresis separates DNA by size, and it moves DNA using an electric field. The smaller pieces, because they get stuck in the holes, travel further down the gel. And the larger pieces travel slower because they get stuck in the holes in the gel. So that way you can separate the DNA fragments by size. The larger ones, the larger meaning more base pairs, are up here, and the shorter ones, meaning less base pairs, down here. So this is another pictorial representation. So here we have our DNA samples, and we're going to pipette them into the well. These are called wells, and then apply a current, and the current will cause the DNA molecules to move down the gel according to size. The longer molecules will get cut up, will stay up top, on top of the gel, and the shorter DNA molecules will make bands towards the bottom of the gel. Once you run the gel runs for about an hour. It's usually a half an hour, but for some reason the gels that we have are slower. So we run them for about an hour. It takes about an hour to separate the bands of DNA, which we'll do on Thursday. Then you need to stain the gel. But you can't, just by running the gel, you can't visualize the DNA. The DNA is actually clear in color. So you have to stain the gel so that you can see the DNA. Kaiser, go away. So real labs, well, not that we're not real lab, but real forensic scientists or real scientists in general use aethinium bromide. It's a much better stain. But aethinium bromide is carcinogenic. So when I did this in college, we would do it under a fume hood with like these big, huge gloves and stain pour aethinium bromide on the gel. And let it sit, and then take it off, and then we would take it to a UV light box, and we'd give pictures like this, which show the DNA bands. Each specific band here, and I don't know why my pointer keeps going away, each specific band here represents or corresponds to a strand of DNA of a specific size. Sort of same thing here. So if you look at this gel over here, which is sort of a pictorial representation of that, the bands on the top are so this is, let's say, one sample of DNA. This is the longest band, and then down here is the shortest band. And each of these is a different fragment of DNA. Some of them, if they line up, that means they're the same size. We will not be using aethinium bromide to stain our DNA. We will be using something called fast blast stain, which doesn't stain it as well, but we're not allowed to use aethinium bromide since it's carcinogenic in the lab. Okay. So now we're going to sort of move into the uses of gel electropheresis. And this is probably going to take 2 videos as I'm running out of the 15-minute time limit, so I'm going to do this in 2 videos, but that's okay. So just to recap, DNA gel electropheresis separates DNA by size based on the number of base pairs. So the largest fragments, they stay on top of the gel, so they would be up here. And the smaller fragments travel all the way down the gel. And the reason why the DNA goes down the gel is that we apply an electrical current. And since DNA is negatively charged, it will travel towards the plus side of the gel. Now, what's the point of this? Besides the fact that when we want to run PCR, you want to see if we got what we wanted. That's one use. So run the PCR, then run the DNA on a gel to see if we actually got any DNA. That's one use of it. But what are some practical uses of gel electrophoresis? And the first one is that it could be used for medical diagnosis. So there's a disease called Huntington's disease, and it causes gradual sort of motor skill loss, and sort of mental capacities get lost over time. It's a progressive disease. It develops over time, usually in your 20s. You don't start developing it until you're in your 20s, and it gets worse and worse as the disease sort of progresses. If you guys remember the show House, which was one of my favorite shows when it first came out when I was a freshman. No, maybe sophomore in college. So 13, she had Huntington's disease in the show where she was being tested, and House sort of like got the test done without her knowledge, or so they sent, you can get this test done to see if you have it, and House like stole some of her DNA from a Diet Coke can, I think it was, and got her tested to see if she has Huntington's disease without her knowing. Well, Huntington's disease causes gradual deterioration of the nerves in the brain, which then slowly but surely kills you because you lose your brain function. The mutation caused by Huntington's disease is the Huntington's gene has several CAG, so the letters, the nucleotide CAG, in the gene. So here with this, there are a certain number of CAG repeats in the gene. People with the disease, for some reason, okay, they get more CAG repeats. So maybe a person without the disease has about 8 to 9, the letter CAG continue or replicate in the gene right here. So like a person who has the disease will only have 8 or 9 CAG repeats. But a person who has Huntington's disease will have more CAG repeats. I think it's upwards of between 17 and 20. So if you have more CAG repeats in the gene, that makes the gene longer. So people who have Huntington's disease, their gene for this protein is longer. As a result, if you design a primer that will replicate this part of the gene using PCR, so design a primer here, and this will be the same primer because it's on either side of the gene. So design a primer here and design a primer here. You can then run 2 tubes of PCR. Okay. Now, because people who have Huntington's disease, this gene is longer because they have more CAG repeats. Okay. What does that mean? What will we see on this gel over here? Okay. Well, if this gel over here, this one right here, is the person without the disease. Okay. You'll see, if you look at the banding pattern here, I've actually just messed up, so we're going to go back. So if you look at I'm just not going to use their banding pattern because that's actually testing for something different. So let's go back here. We ran PCR, so we designed a primer that will replicate this part of the gene and this part of the gene from 2 different people. This is the gene that will not cause the disease, and this is the gene that will cause the disease. Now remember, because primers, so we're going to replicate this part of the DNA, because people with the disease, and it's not shown here, have more CAG repeats, their gene is longer. So as a result, someone that has a gene, or has the gene that causes a disease, since their DNA is longer in this region, or the PCR will replicate this area and it will be longer, the band will be closer to the well. Okay? And here, since this is shorter, because it doesn't have as many CAG repeats, and when I say CAG, let me just write it, I mean these nucleotides that are repeat. If they run PCR on this gene here, their band will travel further down the gel. In order to have Huntington's disease, so Huntington's disease is a dominant gene, you only need to have one of the one disease-causing allele. Remember, you get 2 alleles, one from your mother and one from your father. And Huntington's is a dominant disease, so you only need one of the alleles, and then you have the disease. So if you did this and you see that someone had a band up here after running PCR, that would mean that they have Huntington's disease. Okay, so I'm going to run out of the 15-minute time limit. So I am going to load this video now, and then finish in another 15-minute video. We probably have about 5 minutes left there.